Northern Wisconsin. Actually, the middle of Lake Superior. Right in the middle of the O. So, like many students here, Shannon is a polymath. She's brought her research talents to many areas, both here at Lawrence and elsewhere. She's done organic synthetic research with me, trying to make new organic cobalt cancer drugs. She's worked with Dr. Dixon in the Hog department on angiogenin projects. And she's also done research with my old Organic 1 professor, Dr. Stephen Cass at the University of Minnesota, looking at pretty hardcore physical organic chemistry, so reaction mechanisms. So today she's going to talk about the Mimivirus, which I have to admit I assumed was a typo. I assumed that Dave Hall had misprinted the word. It actually was Minivirus, but it's not. I Wikipedia'd it, and it's really a thing. Let's hear her describe it.
Okay, thank you so much. I apologize for a little bit of a late start. Hi, my name is Shannon Kreuser, and today I'm going to be talking about the Mimivirus and its impact on the human immune system. But before we dive in, let's start with what a virus is. Viruses are organisms that are kind of on the edge of life. They're got one foot in the pool of life and one foot out. This is because they can only replicate inside of living cells. Normal cells can divide on their own and make new ones. Viruses can only do that once they're inside of a living cell, then they can make more copies of themselves. But they're completely reliant on host machinery. They also have no metabolism, so they can't make or utilize any energy, and again, they're completely dependent upon the host machinery for that as well. They can't make any building blocks of life or get any energy from the sun or anything like that. However, they do inherit genetic information, and therefore they're subject to evolution just like we are. So they're sort of life adjacent. The basic structural parts of a virus is there's two necessary parts. We have the genetic material, which is just the information that's carried inside of the virus, and the protein capsid, which keeps it safe and helps it enter new host cells. The capsid can either be icosahedral, which is this sort of geometric sphere shape, or it can be filamentous or a cone shape. It depends on the virus. And the genetic material can either be DNA or RNA, which is sort of two different flavors of genes. Some viruses also have this lipid coating on the outside, as you can see the herpes virus does. And this often has receptors on the outside which help it recognize and get into host cells. Some famous examples I'm sure you've heard about is herpes simplex 1, which is the virus that causes cold sores. We have the measles virus, which causes measles. It's actually, there's a pretty large outbreak of it in Ohio right now. There's 86 cases, and it's the largest outbreak since 1996. There's the polio virus, which caused the outbreak of polio, which in the early 1900s causing paralysis in many children. Ebola virus, which if you were listening to the news about 2 months ago, there was an outbreak in Ghana. It causes hemorrhagic fever and is pretty deadly. There's also the MERS coronavirus, and MERS stands for Middle Eastern Respiratory Syndrome, and the coronavirus is just the type of virus that causes it. This is what the CDC is kind of currently worried about, and there's found 2 cases of it in Orlando. So, but don't worry, not all viruses even infect humans. Not all of them are going to kill you. What I'm talking about is Mimivirus, which is this guy right here. So he was found, or she was found, in 1993 in Bradford, England, when there was an outbreak of pneumonia and scientists were trying to figure out what was causing it. So they took some samples from the water tower and cultured it in amoeba and looked to see what would grow in the amoeba. And they found this. They thought it was a bacterium. Now, bacteria are pretty much the rule of thumb is they're 10 to 100 times larger than viruses. So the fact that there was a virus that was as big as a bacteria was, they didn't even consider that it could be a thing. So they just assumed it was a bacterium. Thus, that's why they called it the Mimivirus, for mimicking microbe, since it's a microbe that mimicked a bacteria. In 2003, however, they did further studies on it, and they found that it actually was a virus. It didn't have all the machinery it needed to be a fully living cell, and it replicated like a virus. So this was very much a watershed moment. This pretty much, we had no idea viruses could get this big or be this large and still function. And this pretty much led to a sort of outgrowth of finding other viruses in the environment that are just as large. The basics of the Mimivirus. Its official name is Acanthamoeba polyphagia mimivirus, or APMV for short. It is a giant DNA virus, so it has DNA for its genome. It is icosahedral, so it's that geometric shape. And it's 150 nanometers in diameter. Now let's give some context for that number. Here is HIV. HIV is 120 nanometers, and it's pretty much a typical virus. You can get viruses all the way down to 30 nanometers and all the way up to around 250 nanometers. But here's our Mimivirus, 750. That's a huge jump in size. It's so big that it's even larger than some bacterium that we know of. This is the Mycoplasma genitalia, and it lives inside of other bacterium or other amoeba, and it's only 330 nanometers. And here's our SxmrYqIMgDA, which is the smallest known archaea, and that's only 400 nanometers. And our Mimivirus is larger than that. This is our E. coli, just to give you a regular E. coli cell, to give you a size comparison, which is about 1,000 nanometers wide and a few thousand nanometers long. Now, its genome length is also what makes it unique. It is a huge genome, and the length of a genome sort of corresponds to the complexity of an organism. And our genome for the Mimivirus is 1,200 kilobases. Now, humans have about 3 million kilobases. This is a scale that's a logarithmic scale of the genome length of multiple different organisms. But don't be fooled, it's a logarithmic scale. So each of these hash marks is not just increasing by 100, it's increasing by a factor of 10. So here's our about average virus, the HIV. It's got about 10 kilobases worth of genetic material. Our Mimivirus has 1,000 kilobases worth of genetic material. It's right around the same size as this P. ubiquae, which is the smallest free-living bacterium that we know of. So the P. ubiquae uses 1,300 kilobases of genetic material to freely live, metabolize, divide, and grow. And our Mimivirus only uses 100 kilobases less in order to infect other cells. This really sort of implies a level of complexity that was previously completely unknown for viruses. And as I said before, this discovery of the Mimivirus led to us finding new viruses that are this large. This is because previously when we were looking for viruses, we actually used filters that only had holes that were 200 nanometers large. So we had to find new ways of finding them. And since then, we've found many others in the environment, sort of in water samples or other environmental samples. There's the Mimivirus, which has a genome that's slightly larger than our Mimivirus. It's actually in the same family. It's got the same structure. There's the picovirus, which is physically the largest virus that we know of, at 1,100 nanometers. This virus is really fascinating because it was discovered in an Arctic tundra sample that is 30,000 years old, and we've been able to culture it in amoeba today. We also have the Pandora virus, which has the largest genome that we know of, at 2,400 kilobases. So this is by far the largest genome that we've seen and in a virus, and definitely rivals the genomes of many bacteria we know of. So the Mimivirus replication is also unique. It's so large and so complex that the way it replicates is surprisingly independent compared to other viruses. Most viruses come in, they insert their DNA into the cell's nucleus, and then the cell sort of treats those genes like they were their own and makes the proteins from that, without really knowing that they're virus genes and not their own. The Mimivirus gets into the cell, then makes what we call a viral factory. This viral factory is a bunch of lipid membranes that are bound together, and it has a lot of the Mimivirus machinery in it. And it allows the Mimivirus replication cycle to have a level of independence from the host machinery that we really don't see in other viruses. In fact, it's so complex and so independent that it has a virus that infects it. This is a virus within a virus. We've never seen this before, and we don't know of other ones that exist, but that doesn't mean they don't. We just haven't found them yet. It is called the Sputnik virophage, and here you can see it inside of a Mimivirus particle. It's much smaller, it's only 15 nanometers, and it's only 18 kilobases worth of DNA. Like I said, it uses those viral factories to replicate more of itself, and it actually causes the Mimivirus to become malformed. Now, the natural host of the Mimivirus is the amoeba. That's what we found it in, that's what we cultured it in. But many viruses have lots of other hosts. Influenza A has, its natural host is birds, but it can also infect humans. If you've ever heard of bird flu, that's what that is. And that's what this is. also, the cucumber mosaic virus is a virus that infects up to 1,000 different species of plants. So viruses can be very generic in the number of different organisms that they infect. So we want to know if Mimivirus infects other cells besides just amoeba. And what better place to start than with ourselves? Also, the reason we ended up looking at human cells is because when we were first finding it, we were looking for possible pathogens that caused pneumonia in the environment. So this is sort of a good place to start, to see if it causes pneumonia. There's actually work papers that in 2007 and 2005 hinted at this and gave some evidence for the fact that Mimivirus does cause pneumonia. Antibodies were found against Mimivirus in patients with pneumonia. Now, an antibody just means that the patient was exposed to a Mimivirus at some point. It doesn't necessarily mean that that is what was causing the infection, but the fact that the antibodies were not found in healthy subjects really gives a clue that the Mimivirus was the one causing the infection and not just an environmental virus that was hanging out. So the antibodies are part of our immune system. They're part of the adaptive immune system, which acts slower but is more specific. There's also another part of our immune system, which is the innate, and it acts much faster and is very much mediated by cells. Now, viruses want to stop the immune system. They want to be successful in infecting us. So what they'll do is they'll often infect or disable macrophages. This is because macrophages, they sit in between the innate and the immune systems. These are large, very large white blood cells, and what they do is they phagocytose or eat pathogens. And we know that the Mimivirus can get in via phagocytosis, but we want to know if it can successfully replicate within those macrophages. So that's the next question. And in 2008, we got some data that actually supports this. What they did is they infected macrophages with Mimivirus, and then they looked to see the number of copies of the DNA viral genome. So here on the y-axis, we have the number of DNA copies, and on the x-axis, we just have time, 0 and 30 hours. And you can see a significant increase in the number of copies of the genome, indicating that the Mimivirus is actually replicating and making more of itself actively within those macrophages. Now, if the Mimivirus can infect these macrophages and is interacting with our immune system, it opens up a whole new host of questions for how exactly does it interact with our immune system, what does it have the ability to interfere with, and what does it respond to? Now, viruses and host immune systems often have this sort of complex interaction. It's called host-virus interactions, and this is where the immune system finds a way of stopping a virus, and the virus finds a way around it, and it goes on, and it's really sort of an evolutionary arms race, where I've got a bigger gun, now you've got a bigger gun. Whenever we see this sort of complex interaction, it really means that there's a shared evolutionary history of infection. So this virus has been infecting this type of cell for quite a while. And we really want to see if that's been happening with Mimiviruses and macrophages. So we're going to be focusing on the interferon system, which is part of our immune system. Now, interferon is an antiviral signaling protein. It shuts down protein synthesis. So with normal DNA viruses, it can't co-opt the host machinery and make those proteins as easily. However, it also stimulates and turns on a bunch of other more specific antiviral genes as well. You may have heard of interferon because it's used as the first form of treatment against MS as well. That's really its most common, it's the most pop culture reference for it. We're going to be focusing on the type 1 interferons, which is the interferon alpha and interferon beta, and this is interferon beta right here. So let's talk about how interferon gets produced. So here we have a human cell, and our virus has entered our body. Human cells have what we call PRRs, or pattern recognition receptors. What these are receptors that can recognize certain molecular patterns that are very common on different types of viruses, and they can bind to them. So when they bind, they activate the receptor. This receptor turns on. Now this tells the cell that, hey, something's wrong, there's an intruder, we need to do something. So we're going to signal the alarm by turning on interferon. But interferon is not just acting within the cell, it's acting without. So it needs to signal its neighbors, so it needs to be released. That's really its goal, is to tell the neighbors, hey, something's going on, somebody's invading, we need to get ready. So interferon is coming in and meeting a new cell. This adjacent cell has what we call an interferon receptor, and that's just something that recognizes the interferon and binds to it. And when it binds, it becomes activated. Now, interferon alpha and interferon beta both bind the same receptor, but they end up having slightly different cellular responses. And we'll talk about that later. Once the interferon receptor is activated, it tells the nucleus that we need to start preparing for a viral attack. And so what it does is it produces these ISGs, or interferon-stimulated genes. These are a bunch of different proteins that have specific different antiviral effects and keep the virus from replicating too effectively within the cell. Two that we're going to be talking about in the paper later on are MX1 and IFI6. MX1 inhibits replication of viral DNA, and IFI6 plays a role in regulating apoptosis. Apoptosis is just programmed cell death, and sometimes it's important to kill a cell before the viral infection reaches the point where it's going to kill the cell on its own. So it's a way of curbing the infection. Now, viruses don't want to be stopped. They want to be successful. So they're going to try to find ways to fight back. And there's a couple of different mechanisms that they do this with. First is what we call viroceptors. Viroceptors are these soluble proteins that viruses release out into the cell when they're actively replicating in a cell. And what they do is they bind up interferon. They basically put a gag on it so it can't signal the adjacent cells and can't sound the alarm. This is an example of a virus that successfully produces viroceptors against interferon alpha and beta. It's called vaccinia virus, Western Reserve, and it's actually what causes cowpox. And because it's so physically similar to smallpox, this is what we actually used in the first smallpox vaccines. Another way that viruses can interfere with the production of these ISGs is inside the cell. They can either stop the signaling from the receptor to the nucleus or stop the production of the proteins from the nucleus to the actual final product. This is a little bit harder to look at though, since there's so many complex systems within the cell. Now, so if Mimivirus successfully replicates in human cells, does it interact with our immune system? That's what we really want to know. But within that question, there are some sort of smaller questions. We want to know if when macrophages are infected, they actively produce interferon as a result, meaning that they recognize the Mimivirus as a pathogen. We also want to know if ISGs are produced. We want to know if that interferon is successfully accomplishing its goal and getting the antiviral response ramped up. We also want to know if the Mimivirus possibly makes viroceptors. It's such a complex virus that it's very likely, since other complex viruses produce those viroceptors. We also want to know if the interferon is at all effective against the Mimivirus in halting that replication process. So the paper that I'm going to be discussing is called A Resourceful Giant, APMV is able to interfere with the human type 1 interferon system. And again, APMV is just a shortened version of Mimivirus. And this is by Lorena Silvia, and it was published in 2013 from the University of Minas Gerais. So the basic methods of this experiment were to collect macrophages from blood samples from human subjects. Don't worry, the humans were voluntary. Nobody was taken advantage of. The macrophages were then infected with Mimivirus, and from those cells infected with Mimivirus, we isolated the RNA from those cells. Then we ran PCR, which is just a method of amplification to make one copy of RNA into hundreds of copies. And we looked to see if interferons or ISGs were being produced. So some of the controls that we used in the experiment are parainfluenza virus 1 Millhill strain. Now, if any of you know what the croup is for babies, this is what causes that. And this is a virus that we know successfully stimulates the interferon system, so we're using this as a positive control. We also have UV inactivated Mimivirus, which means that it cannot replicate, like the machinery and the genes within the Mimivirus are completely inactivated, but it still has the same structural coding, so those PRRs, those pattern recognition receptors, can still recognize it. And whenever you see mock, it just means that the same procedure was followed, but no virus was added, so that's our negative control. Now this is the first set of data. On the y-axis, on the x-axis we have hours post-infection, and on the y-axis we have the fold induction of the interferon alpha mRNA. So this is just looking to see how much interferon alpha was produced as a result of infection in the macrophages with Mimivirus. Here is the cells that were infected with Mimivirus. Here's our positive control, and here's our negative control. As you can see at hour 18, there's a significant production of interferon alpha by the macrophages. And when they did the same experiment with interferon beta, again we have hours post-infection and the fold induction of the interferon beta mRNA. We can see that at hour 18, again we see this significant production of interferon beta. So what this really means is that these macrophages, these human cells, are recognizing the Mimivirus as a potential pathogen, and they're sounding the alarm. They're effectively getting the signal out there that, hey, something's wrong, we need to respond. The next question is we want to know if the interferon is successfully leading to the production of ISGs, or if possibly the Mimivirus is interfering with that in some way. And so they did pretty much the same experiment. They infected macrophages with Mimivirus and a couple of different controls, and then they looked to see at the production of the mRNA. In this case, they were looking at MX1. And MX1 is one of those ISGs I talked about earlier, which inhibits viral replication. Now, here we have hours post-infection, and on the y-axis we have the fold induction, and here is the Mimivirus data. Now, there's almost no MX1 production for the active Mimiviruses, and this is the same as our negative control. However, the UV inactivated Mimiviruses, the ones that can't replicate but can be recognized, have a significant portion of MX1 being produced and match our positive control. When we did the same experiment, we looked at the IFI6 mRNA. They, again, is hours post-infection, but here we were looking at a different ISG to look at the production of that ISG. And again, this is the Mimivirus data. Here's our negative control. There's no IFI6 being produced, or practically no IFI6. Here's the UV inactivated, again, the Mimivirus that cannot replicate but can be recognized, and our positive control. So this is kind of interesting because what this means is that the Mimivirus is being recognized and the interferon that's being released is being successful when the Mimivirus can't replicate and can't produce its own proteins. But there's something in the active Mimivirus, something that's being produced when the Mimivirus is being replicated, that is stopping that signaling process. It's one of these mechanisms. We don't know which one. Something is inhibiting the production of these ISGs, and some of them are not being produced. So we want to know how. That's really interesting. We want to know what mechanism it's going through. One of the easiest ones to look at first is the production of viroceptors, since they're outside the cell and a little bit easier to look for. So they ran an experiment where they infected infected macrophages with both Mimivirus and the vaccinia virus, which we know produces viroceptors. They then took the liquid from those cultures and assessed the activity of the interferon in the solutions. Now, if there's no activity, that means that the interferon's being bound and cannot signal anybody. But if it's free-floating, then it will work. So here is the interferon alpha data, Mimivirus and our vaccinia virus. There's no interferon activity for the vaccinia virus, and there's a significant amount of activity for the Mimivirus. And it's the same for the beta. So this really indicates that no viroceptors are being produced by the Mimivirus, since all of the interferon that's in the system is free-floating. So that means that something is going on within the cell. It's interfering with the production of the ISGs in a mechanism that's inside the cell and not outside of the cell. So since some of these ISGs are not being produced, we want to know if the interferon is affecting the viral replication at all, or if it's just not effective against Mimivirus. So what they did is they pre-treated macrophages with different concentrations of interferon alpha, and these are these samples right here. And this sample was not pre-treated at all. Then they infected the macrophages with the Mimivirus and looked to see, after a certain number of hours, how many viruses were produced. This is the viral titer, which just means the concentration of viruses in the solution. there's no real significant difference between these different data points, indicating that the interferon alpha doesn't really have a significant impact on the viral replication of the Mimivirus. They wanted to do the same experiment but looking at interferon beta, and they did, and they saw a great reduction in the number of Mimiviruses that were produced. So again, we have the different samples treated with different concentrations of interferon beta, and this is the viral titer. And there's a huge reduction indicating that some of the ISGs that are produced by interferon beta are being successful against the Mimivirus replication. So some of these are working, but the ones that are produced due to the activation of interferon alpha are not really making a difference. This is kind of fascinating. So some of the conclusions that they found in this study are that Mimiviruses do actually infect human macrophages. So they're actually replicating inside of us, well, if you're infected with them. And the fact that it does interact with our immune system pretty complexly. It not only activates the immune system, but it also inhibits some of the production of the ISGs as a result of the interferon. It also does not produce viroceptors, but it is negatively affected by some of the ISGs produced by interferon beta, but not interferon alpha. So this really complex interaction between the Mimivirus and our human interferon system means that Mimiviruses and macrophages have probably been infecting and interacting with each other for quite a while, and we just didn't know that they were a thing because we didn't know viruses could get this big. Some future questions to look at as far as the Mimivirus and human immune system is how exactly does the Mimivirus interfere with this ISG production? What is going on inside of the cell that's stopping this? And also, which exactly are the ISGs that are produced by interferon beta that are effective in halting the Mimivirus replication? I'd like to say thank you to Dave Hall for helping me understand viruses and helping with this talk a lot. Allison Fleshman listened to me a couple of times. Kim Dixon, who's my advisor and helped me a lot along the past few years. And Stephen Deverett, who I also did research with and was a huge help. Thank you. As far as we know, and we've been able to visualize that it actually co-localizes to those viral factories and is replicating inside of those viral factories. Yep.
I had a question about the viral factories.
Yep.
Do you know if the Mimivirus brings in all this machinery with it, or has to hijack the cells stuff to build the factories?
So that graphic is a little bit inaccurate in the fact that it says that the DNA never goes into the nucleus. There is part of it where originally, some of the DNA goes into the nucleus and some gene products are made, but from there the viral replication is pretty much located in the viral factories.
Cool.
Mm-hmm. Any other questions about anything? Yeah?
How did you become interested in this topic? Because I know you did a lot of research in a bunch of different other areas.
Yeah, so I did a tutorial with Dave Hall last term and Mary Schultz and Emily Hoylman and Nick Waldner on viruses. And there was a paper talking about these giant viruses and how they're sort of redefining our, their level of independence and complexities, kind of redefining our definition not only of viruses but also of sort of microbes and life. And this Mimivirus was the first one to be found, so it's definitely the one we know the most about. Like the picovirus and the Pandora virus, we've only found within the past 2 years. Yep.
Slightly biochemistry ignorant here, but you said it seemed the main point was that there's been an evolutionary history between humans and the virus. But could there be some sort of a convergent evolution type mechanism that would lead to the same type of response?
Yes and no. I mean, it's probably possible for some of these to be convergent evolution, but some of the complexity of the interferon system and how complexly the Mimivirus is really interacting with it, the fact that it does stop that production at some point. The amoebas do not have interferon systems at all. They barely have an immune system because they're single cells. They do. But they do not produce interferon at all, and so that whole signaling pathway is entirely different. Yep.
So, since they found the Mimivirus, you said they found a whole bunch of other
Yes.
viruses that are the same
Mhm.
size. Are there other groups doing the same type of analysis to see if it's infecting humans as well on the other big ones, or is this Mimivirus kind of leading the way in this new field of large viruses?
It's definitely the one we know the most about. That doesn't mean if there are other groups looking at it, I'm not sure they're publishing yet. But yeah, like I said, the Pandora virus and the Pithovirus, which are those two really large ones, were only found in the past 2 years. So, but definitely the megavirus, and there's another version of the Mimivirus that's in the same suborder, and it's called the Mama virus because it's slightly bigger. And they've been doing some studies with that. Those are, I think, it's a little bit less conclusive, but they're kind of everywhere. Like they found some of these viruses just like in water samples in Chile, and like I said, the Pandora virus was just in a soil sample in the Arctic tundra, and they've been finding antibodies against these the Mimivirus in like just cattle and macaques in the Amazon. So this whole environmental niche that we have not really paid attention to because we didn't know it existed. Right.
Thank the speaker again.